In this video we will discuss about CD8 lymphocyte activation. That is one of the important lymphocyte that during that plays an important role in immune system. So there are two different situation that has been shown in this figure by which CD8 plus T lymphocyte gets activated. We will go through both of this figure one by one. Let us go through the panel A, in which the virus infect the cells. When the virus infect the cells, it contains either its genetic material either in DNA or in RNA. If the genetic material is RNA, it also virus package also include reverse transcriptase that helps in uh, translation of or reverse translation of this RNA into the DNA that can integrate with the host. However, in this case, it is shown that the RNA has been directly translated in the cytosol to form a viral product, or to form the product that is encoded in the genetic material of virus. And proteasome is usually present in the host cells. And what happens is this proteasome will digest this viral protein, some of the viral proteins, and convert them into the peptide, and then these peptides interact with the TAP that is usually present at the endoplasmic reticulum, and this TAP helps in binding the antigen of the viral protein to the MHC1, it is major histocompatibility complex 1. And this after that the major histocompatibility complex 1, with the help of or follow the secretory pathway via Golgi, then uh, express itself along with antigen at the cell surface. And this antigen, this major histocompatibility complex, then interact with the immune system to share this antigen that helps in the activation of CD8 plus T lymphocytes along with other immune response. The next panel shows uh, another situation for activation of CD8 plus T lymphocytes. When some of the cells that are infected with the virus and are dead, uh, and some part of those cells, that is cell debris with viral protein, interact with another live cells, and and in case this some of the cell debris come inside the cell, then also that also helps in the activation of CD8 plus T lymphocytes. But there are different ways by which CD8 plus T lymphocytes can be activated in this case. First one is the same process as it occurs in the first case, that is the translated viral protein gets digested with the help of proteasome, and then this peptide interact with TAP that is present in the endoplasmic reticulum, and that helps, and the antigen of this viral protein then interact with the major histocompatibility complex 1, and this major histocompatibility complex follow through the Golgi follow the secretory pathway through the Golgi and express itself on cell surface level. And another approach is that instead of following the secretory pathway through Golgi, the major histocompatibility complex along with antigen interact with phagosome, and that helps in transferring of this major histocompatibility complex or MHC1 to the cell surface level. And third way is that the translated protein directly interact with proteasome or lysosome proteolysis, that is cathepsins, and that does the same function as it is done by proteasome, that is digest the viral proteins, and some part of and the peptides interact with the major histocompatibility complex that then express itself at the cell surface level. So in this case there are three ways by which CD8 plus TV cell can get activated. So now let us understand what are the additional signals that helps in increasing the signal of CD8 plus T lymphocyte or CTL, that is cytotoxicity T cell lymphocyte. in the previous video I was not able to tell you the full form of CTL, in one of the previous video. So full form of CTL is cytotoxic T cell lymphocytes. So uh, how this how the signal of this T cell lymphocyte increases. It increases if it receives the following signal, that is interleukin 2, interleukin 12, interleukin 21, interleukin 27, and this AG signal. I'm not able to recall the full form of AG signal, but you can go through the reference that is provided in the description for more detail. So what happens after that, these signals are received by CTL. It will prevent the MPEC TCM cell differentiation. These are the memory cells, and these cells have the memory of the antigen of the virus. So by preventing this cells differentiation, uh, uh, it helps the it increase the life of MPEC and TCM cell, so that helps the host immune system to fight with the virus in a more efficient manner. And third one, and the next one is the effector function. It will improve the other immune pathway, that is IFN alpha TNF, INF gamma TNF alpha, perforin and granzyme. We have discussed perforin and granzyme role in one of our previous videos. You can go through that. And third, that is, it helps in the migration of CCR5 and CCR7. As we know that the entry of HIV virus requires CCR5 as a co-receptor. So by affecting this CCR5 or migrating this CCR5 CCR7, we will prevent the entry of the some of the virus that requires these kind of co-receptors for their infection. Third one is the last one is the or last main point that increases the exhaustion. So what happens in that case? In case of chronic diseases, uh, it is essential that the CTL level always maintain at a at least a minimum level, so that uh, uh, virus does not uh, reach to a point that it is uncontrollable. It is CTL level should always be at least minimum, so that virus does not get uncontrollable. So for that case, this exhaustion pathway plays a role, as takes care it will takes care of the minimum CTL level that are present within the body. However, I might be wrong in understanding this point. You can go through in detail in the reference that has been provided in the description. Last part is that CTL cells that are present near the infected tissue get some additional signal, that is interleukin 2 from CD4 plus T cells and IL-27 from innate cells, and this AG signal from infected cells. So what happens is that it will release this interleukin 10 signal, and the interleukin 10 signal released from CTL helps in dampen inflammation. That is, it helps in controlling the inflammation and tissue damage at the site of infection. So these are some of the points of CD8 T lymphocytes activation, and how its signals can be increased, and what are its implications. So in order to understand all these things in details, you can go through the references that are provided in the description. That's all for this video. Thank you for your time.